Welcome. In this video we are going to discuss about transduction. Now the transduction is also known as virus mediated transfection. Okay, so transduction is virus mediated gene transfer. Now there are certain animal viruses which naturally infect human cells and mammalian cells. Now these animal viruses include both DNA viruses as well as RNA viruses. DNA viruses example is SV40 or adenoviruses, and RNA viruses example is HIV or retrovirus. So with the help of these viruses we are going to introduce our gene into our target animal cell, and that is what transduction is all about. So modified form of these viruses are known as vectors, and these vectors made from these viruses can be used to transfer the exogenous genes into a suitable target cells at high efficiency. You can modify this animal virus, you can make vector out of it, and then you can use that vector to transfer exogenous genes into suitable target cells at high efficiency, and that is what transduction is. Now within the cell, some of the viruses replicate episomally and other viruses integrate into the genome of the host organism. Episomally, it means it will exist as extra chromosomal DNA or RNA, and it will not depend upon the chromosome to replicate. It can replicate on its own. That is what episomal means. And in the other condition, the virus genome will integrate into the chromosome. So as the chromosome replicate, that integrated virus DNA or RNA will also integrate. Now the development of these viral vectors for DNA transfer into the animal cell have advantageous characteristics of animal viruses. Okay, animal viruses have some characteristics which are advantageous to use them as a vector for DNA transfer. Now what are they? Let's see. Now they have evolved, these animal viruses have evolved efficient mechanism to get adsorbed on the cell surface, and they enter the cell without damaging the cell. Okay, that is one characteristic which is very useful when you are making virus out of it, vector out of it. And the second advantage is that the viruses deliver nucleic acid intact into the cell. And why is that? Because the nucleic acid is initially packed into proteinaceous capsid. Okay, so the DNA which is getting integrated, that DNA or RNA is in the intact form. Now viral genomes have strongly regulated elements, and these elements of the viruses can be exploited for high level of foreign gene expression. So viral genomes have some elements. If we exploit them, then we can achieve high level of foreign gene expression, and that is another advantage. Many animal viruses have broad host range, and that is the reason why they can replicate in diverse cell types. Okay, they have a broad range, and in many different cell types they can replicate. Now many viruses stably transform the cell, either by integration or by latent episomal replication. Okay, so you get stable transformation of cell if you are using viruses. Now virus mediated transfection have some drawbacks as well, and the major drawbacks are immunogenicity or cytotoxicity. Okay, these two are the major drawbacks of this virus mediated transfection process. Now this introduction of viral vector can cause inflammatory reactions and insertional mutation. Because of the fact that the viral vectors randomly integrate into the host genome, they might interrupt the essential genes. Okay, so they can cause inflammatory reactions or insertional mutation. And if they randomly integrate into the host genome, and that is the reason why they can interrupt an essential gene of that particular host organism, which might affect the survival of that particular host organism. And another disadvantage of using viruses as vectors for transfecting the animal cells is that viruses have limited space for packaging the foreign DNA. So you cannot transfect your animal cell with a very large sized DNA. There is a limited space and you have limitations on the size of the foreign DNA which you can insert into the viral vector. So that is another disadvantage. So that is all about transduction. Thanks for watching and see you next time.